Hello everyone. Today I would like to discuss about the topic plant tissue culture. So from this topic you will understand the following aspects. One is what is plant tissue culture? What are the types of plant tissue culture? What are the requirements of plant tissue culture and how to establish plant tissue culture? So what is plant tissue culture? So plant tissue culture is nothing but in common terms it is known as in vitro cultivation of the plant cell or the tissue under aseptic and controlled environmental conditions in liquid or on solid media which consist of well defined nutrients for the production of secondary and primary metabolites or to regenerate into a new plant. So the success of this plant tissue culture is depends upon the inherent capacity of the plant cell, which is known as totipotency. So here the totipotency is described in two terms. One is dedifferentiation and the redifferentiation. So what is dedifferentiation and redifferentiation? So here the explant is taken from any part of the plant. It may be leaf, it may be root, stem, etc. The explant is taken and it is inoculated on solid media or in liquid media. So this explant, which is an differentiated tissue, undergo dedifferentiation. That is, it undergo meristematic stage, which is nothing but actively dividing cells by utilizing the nutrients from the liquid or the solid media. So which give rise to an undifferentiated, unspecialized mass of the cell, which is generally known as callus. And this callus, when it is inoculated on particular medium, it give rise to a plant. So it is called as redifferentiation. So the dedifferentiation and redifferentiation is nothing but totipotency. So totipotency is the potential inherent capacity of the plant cell or tissue to develop into an entire plant. So it indicates all the information which is necessary for the growth and reproduction of an organism is contained in the cell. So dedifferentiation is the capacity of the mature cells to return back to the meristematic condition and development of the growing points. So what is competency? Competency is to develop into a plant. So Gottlieb Heberlein, who is the father of plant tissue culture, he proposed the concept of totipotency. He succeeded in the culturing of the leaf mesophyll tissue. So next, what are the types of tissue culture? So based on the medium used, there are two types of the cultures. One is callus culture and suspension culture. So callus culture is inoculated it is done on the solid media, whereas suspension culture is done on the liquid media. So based on the explant used, there are different types of tissue culture: root tip culture, leaf or leaf primordial culture, shoot tip culture, complete flower culture, anther or pollen culture, ovular embryo culture, and protoplast culture. So here the root tip is used, leaf primordial, shoot tip is used, and the anther and pollen grains. So these are all the growing tips of the plants, that is, they consist of the meristematic tissue. So such explants are used for the tissue culture. Actually, any part of the plant is used for the tissue culture, but the tip of the plant, the tip of the leaf, tip of the root, tip of the stem usually used as they consist of actively dividing cells, nothing but meristematic cells. So here you can see the pictures of different types of cultures: callus culture, which is on the solid media; suspension culture in the liquid media. These are the pollens which are used for the pollen culture, and ovular, root tip culture, shoot tips, protoplast culture, leaf primordial culture, and flower culture. What are the basic requirements of plant tissue culture? So following are the basic facilities are required for the success of the plant tissue culture. Equipment and the apparatus. The equipments which are used for the plant tissue culture, mainly it includes hot air oven, which is used for the sterilization of the glasswares, incubator to incubate the tissue culture bottles, and laminar airflow chamber, so without which the tissue culture cannot be carried out. So this laminar airflow cabinet or the chamber is mainly used for the aseptic transfer of the cultures. So along with this, it also needs a separate facilities for the washing and storage, for the preparation of the media and the sterilization room, aseptic chamber for the culture, and culture rooms or the incubator equipped with the temperature, light. Some of the cultures require the light, some of the cultures does not require the light. So accordingly, these facilities or control devices are equipped in the culture room. And it also requires the recording area which is equipped with the computer for data processing. So this is the tissue culture laboratory. Here you can see the culture bottles which are in the slant position. So none of the culture tube in the straight position, as the slants give rise to a more surface area for the culture. So these are the different tools and which are used for the preparation of the explant: scissors, needles, forceps, knife, and these are the glasswares which are used for the preparation of media: beakers, conical flask, funnel, ropers, measuring cylinder, graduated pipette, etc. So how to establish plant tissue culture? So it mainly involves the following steps: collection of the plant and collection of the glasswares, tools or the vessels, and their sterilization, and collection and sterilization of the glasswares, tools and the vessels, and then preparation of the explant, surface sterilization of the explant, incubation on the inoculation on the medium, production of the callus from the explant, proliferation of the culture, subculturing of the callus, and suspension culture. So explant preparation. So the first step is collection and sterilization of the glassware. Already we had discussed about the glasswares which are required for the tissue culture. So explant. So what is explant? So as I told in the beginning, explant is nothing but it is a part of the plant body which has been taken from the plant to establish culture. So it may be taken from any part of the plant. It may be root, it may be stem, it may be flower, it may be fruit, it may be leaf, it may be floral parts, or that is like anthers, stamens, etc. So here in this picture you can see the preparation of the explant. So here we cannot use the whole plant for the culture. So explant should be prepared such that the length of the explant should be around 1 to 1.5 centimeter. So after the preparation of the explant should undergo surface sterilization. So what happens if surface sterilization is not done? So there is a chances of fungal attack. The success of the plant tissue culture mainly depends on this mainly depends upon the cultures without the fungal growth. So if there is a fungal growth and there is no formation of the callus, there is no growth of the plant or any process which involves the plant tissue culture. So because of which the surface sterilization is carried out. Different chemicals are used for the surface sterilization: chromic acid, mercury chloride, which is generally used for the leaf, calcium hypochlorite, which is used for stem and root, sodium hypochlorite, which is generally used for the stem, root, and alcohol is generally used for all the type of the explants. So here in the procedure of the surface sterilization, after the preparation of the explant, first it is treated with the alcohol and then it is washed with water. Then it is transferred to the sodium hypochlorite. Again it is cleaned with sterile water. Then it is inoculated on the media. So inoculation of this explant should be such that the half of the part of the explant should be inside the medium. So what will happen if the complete explant is merged in the medium? So here there is no space for the growth of the tissue. So the media does not allow the growth, because of which the explant should be exposed half inside, half should be exposed, and then half of the explant should be inside the medium. So next is the production of callus from the explant. So now the sterilized explant is transferred aseptically onto the defined media. It may be liquid media or it may be solid media depending upon the type. Then it is those bottles which are inoculated are transferred to the BOD incubator where temperature is maintained around 25 degrees Celsius and light is necessary for the callus production. Then the callus is grown in between the days of 3 to 8 days. So once you get the callus, that callus should be proliferated. That is to increase the amount of the callus, which is done on the proliferation media. So this proliferation media mainly consists of the growth hormones along with the basic requirements. So for the proliferation, the callus is cut into small pieces, then transferred to the media, then allowed for the growth in the callus. So after the 15-20 days, we will get the more amount of the callus. Then it is very important for the callus. Subculturing is very important for the callus because if the callus is left in the medium, so it make utilizes of all the nutrients present in the media, and then there is no media or nutrients available for the explant for the growth. So if it is left like that, the callus or the tissue which is grown will die off. So because of which, to maintain the cultures, the subculturing is done. So subculturing is done after the sufficient growth of the callus. It should be periodically transferred to the fresh medium to maintain the viability of the cells. This subculture will be done at the interval of 4 to 6 weeks. So after the maximum growth, transferred into a potting soil under the required condition. Or it may be used for the production of primary and secondary metabolites. So after the sufficient growth of the callus, so callus is used for the initiation of suspension cultures. So here the suspension culture, it may be initiated by using the explant itself or by using the callus. So generally these suspension culture is initiated using the callus culture. So callus culture is inoculated in a liquid medium and then callus is transferred into the liquid medium and then they are agitated continuously, because of which there is separation of the cells and then they are allowed to grow, and after the sufficient growth they are cultured on fresh media to maintain the suspension culture. And this can be achieved with the help of the instrument which is known as equipment which is known as rotary shaker at the rate of 50 to 150 rpm. Thank you. In next class we'll discuss about the growth measurement of callus and suspension culture.